This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1195, recorded on February 21st, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. Like everybody. Let's see here. I'm sorry, I'm behind on the weather. I'll get it here in just a second. It's freezing, basically. We're going through another cold front, and I've got it right here, 37 degrees. And it was down in the 20s last couple of nights. We have one more day of freezing, and then that's probably it for this winter.
-6 here in Chelsea. Also joining us from Montreal, Canada, Angela Mingarelli.
Hello. So it's also freezing here, a little bit more freezing. It is 17F and -8C, which for us is very warm, considering the last 2 weeks has been like -20 almost every day. So we're happy about it. And tomorrow it's supposed to go up to -1, which is, I think, like 25, something like that. It's exciting.
We sent Angela an Instagram video of a plow in Montreal pushing a big pile of snow, and in front of it there was a car.
Yeah. But the snow was like that. So those of you that don't know, which is possible, Montreal had a record snowfall since 1941. We had 79 centimeters of snow in 24 hours. And even though we're well prepared for the snow, it was still chaos. Like the sidewalks just got cleared yesterday, almost all of them, because before we had to walk on the road. The cars were trying to drive by. It was a little bit of a disaster, but it's starting to slowly melt a little bit, and we're okay.
I remember in Buffalo walking to work and back with Niles on top of the snow piles that were on the side of the road, looking down on the roofs of cars.
Oh my gosh.
You guys need to check out the pic a couple of weeks ago, the Japanese snow tunnels.
Yeah.
Oh yeah.
It's
right,
great. The videos. So the pic, and then the next week we heard from the reader about how it was done. Sorry.
Kathy, you're not on yet.
I know. But I am.
You're also
Spoiler.
joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it really is freezing. It's 32 Fahrenheit, which is 0 degrees Celsius. So those of you east of me expect a balmy warming phase.
Balmy.
Yay.
Also joining us from New York, Daniel Griffin.
Hello, everyone. I was waiting my turn to tell my snow story. No, it's, same weather as Vincent. We're not very far apart. I actually just got back from an outdoor run because apparently people were commenting that I haven't seen you on Strava, which records my runs. And, I was claiming I was on the treadmill, which may or may not be true. So today I got all dressed up, and in this -6C weather, I went for a run right along the water with the wind blowing in my face. And
Wow.
it's cold. It's cold.
Brave.
And my snow stories were from out in Colorado, where I remember one storm where we got a foot an hour. That's like 20 plus centimeters an hour for you, or per hour for you guys up in Canada, right? So we ended up with 5 feet, right? So that's over 100 centimeters in a day. It was so bad that they closed the interstate. The National Guard was using helicopters to drop food for all the animals. And for about a week, I would cross-country ski to and from the hospital because I was back at that time one of the younger docs, and I would see all the patients from my colleagues who were, just not getting to the hospital in the snowstorm.
Wow.
That's
a great
crazy.
story. Also joining
I
us.
just followed you on Strava. Sorry, I also do Strava and now follow Daniel.
Yeah, so you can see me running by the water in the freezing cold.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here, and it's great to see everyone. It is 35 Fahrenheit, 2 Celsius here in Madison. However, if we are talking snow stories, some of you may remember a few weeks ago when I shared the snowstorm that my parents had in Camden, New York. Camden seems to be getting hit quite hard this winter. And so that degree of snow has not really stopped. They're at over 200 inches for 2025. And some of it came in during the past week. And right now, the photos and videos that I am getting are stunning. I've never seen anything like it. Roofs are collapsing. All sorts of issues are happening. And really, nothing like it is anything I've ever seen. So if you want to see some pretty dramatic snow images, I would check out Camden, New York right now.
From Western Massachusetts, Alan Dove.
Good to be here. And despite being from living in Western Massachusetts these days, I don't have a whole lot of great snow stories. Currently, it's 32 Fahrenheit, 0 C, right at the freezing mark. And that is warmer than it has been in a week. And tomorrow it is actually supposed to break freezing and get up to 35. And we're all very excited because the hardware stores were all sold out of ice melt. And everything is covered in inches of ice because we had that storm system come through that was sleet, then snow, then sleet, then sleet, then freezing rain, then snow, then more freezing rain. And it just layered, and it's been here the whole week. Quite a mess.
And from College Station, Texas, Jolene Ramsey.
Hello, everyone. I'll just say that we have the same weather as Rich, and in the trend of the time, I've decided I don't believe in snow. So there you go.
That's the trend. Yep. So you may notice everybody's here. The whole TWiV team. 1, 2, 3, 4, 5, 6, 7, 8. And the reason we're here is because number 9 is missing. Dickson Despommier passed on February 7th. We had just finished recording a TWiV, and I got a text from Marlene saying that. So we're going to talk a little bit about Dickson, what he was and what he meant to us. We're going to miss him. We're going to do some news, and then we'll go into that. And after that, we'll do a snippet and maybe some email, but probably not email. I'm thinking that we're going to just have time for a snippet. And that's why Daniel is here, of course. Daniel's a member of TWiV, but normally does his clinical update on Thursdays. He's going to join us to talk about Dickson. So first, if you enjoy these science programs, if you'd like to hear real science, not fake science, please support us. We need your help to do this and to continue on. And who knows, maybe we're going to have lawyer bills going forward. But please support us, microbe.tv/contribute. Kathy, what's up with ASV?
Well, the election is open for members. It started February 17th. It ends Friday, March 21st. So if you haven't renewed your 2025 membership, you need to do that by February 28th if you want to be able to vote and get a ballot. And then, as you've been hearing, ASV 2025 is going to be in Montreal, and it's hosted by McGill University, July 14th to 17th. The website is asv.org/ASV 2025. And they've asked me to announce that early bird registration ends May 12th, and current members receive a discount on their registration rate. So 2 reasons to re-up your membership so you can vote and so you can get that discount when you register.
I don't have to renew my membership. Right, Rich? You don't have to either.
I don't have to renew mine.
That's great. Good feeling. All right, I'm going to start off talking about Dickson, and then you can chime in anytime, but we'll go in the order of which you joined TWiV and therefore encountered Dickson one way or another. I just want to tell you that on the office hours, our Wednesday live stream 2 weeks ago, we had a nice tribute to Dickson. And let me read this letter from John, who describes it really nicely. I'm sure Vincent will tell all of you about the outpouring of support and condolences on office hours tonight, but if you have time in your busy schedules, I think it's worth taking a moment to view it. Many showed up, regulars and more, and they all had incredibly kind words to say about your dear friend. Many, like me, associate his voice with a compassion and intelligence during difficult times. And I'm sorry for your loss, which you probably feel most directly. Thank you all for every episode and effort you do to make such excellent programming happen every week. All of you have become very dear to your listeners, and I hear you read letters to the show frequently acknowledging and thanking you. And it is because you make a difference and impact our lives for the better with education and intrigue that is nothing less than infectious when presented your way. Cheers and condolences. It was a very nice office hours. I started and I announced that Dickson had passed, and then everyone started talking about him, and that's all we did for 2 hours. It was really great. So I met Dickson in 1982 when I came to Columbia. He'd been at Columbia for many years, where he taught parasitology to medical students. And relevant to the podcast, when his laboratory closed, he worked on trichinella for many years. His lab closed in 1999. He started a medical ecology course, right? How the environment impacts human health. And that was inspired by René Dubos, who was at Rockefeller and he had known during his time there. And so this is what he told me. This is not in his obituary, but this is what Dickson told me years ago. He said he asked his students to determine how much rice you could grow on the rooftops of New York City buildings. And so they did the calculation. They figured out how many buildings there were in the area and so forth. And it turned out to be pitifully little rice. Would not feed the population of New York. So then they had the idea of, well, why don't we take over a building and convert it to a farm? And so he acknowledges that this was a crowdsourced idea. The class came up with this idea, which the class expanded on every year. He wrote 2 books, Vertical Farm and The New City, and kept teaching about it. And of course, ended up doing a podcast with me about it. So he started TWiV with me in 2008. So the first episode, I asked him to join. I had this idea to do TWiV, and I said, who am I going to get? And I said, Dickson. He's a great speaker. He was a great teacher. So he agreed right away. I remember I was walking down the street, and I said, Dickson. And he always go, yes, Vincent. I want to do this podcast. And he agreed right away. So I started TWiV in 2008. Then a year later, we did TWIP. And then in 2014, we did urban agriculture. And he was last on TWiV in 17th of November, TWiV 1167, and TWIP 6th December. And I want to just remind everyone, for the first 20 or so episodes of TWIP, Dickson gave lectures on individual parasites. And then sometime later, after Daniel joined TWIP, which I think was in 2015, they both taught an online parasitology course. And they're both really good resources. And so a couple of other websites I want to mention that are really good. He has a Wikipedia page, and the picture on it I took of him in the subway. We were going somewhere, and he said, Vincent, I need a picture. So I took a picture. I think it was, the post says 34th Street on it, but I don't remember. Then we'll put a link to his obituary. Now, I want Kathy to tell me, they say they have the pronunciation guide, right? Depommeeyay, but that's Despommier, right? He used to say his name Despommier.
Yeah, I think the DE is
Day.
ambiguous when, they're not doing it in IPA, so it's
Yeah.
hard to know.
I would have written Day, but it's a Despommier. He always used to say Despommier. And he has many websites, which you may not know about. You know about trichinella.org and thelivingriver.org, which I cite all the time. The Living River is about stream ecology. Then he has fordomopolis.com, where his students used to make indoor farm plans and post them. Despommierphotoart.com, where he put a lot of his photographs from traveling all over the world. Really nice. I know this because I manage all of them for him. And now I'm going to have to keep up with them.
So the websites are not going away.
They're not going to go away. I have to figure out when the credit card goes to be charged what to do. We'll have to figure that out. Microbe TV can take that over. But they're all really good resources. And so I I just want to say that he was a great colleague. I knew him for many years. I worked with him on all these podcasts. The indoor, what was it, urban agriculture? We did 30 episodes or so. In addition to TWiV and TWIP, he was just a wonderful guy, very nice guy, great knowledge, always curious, um, always game. If you asked him a question, I always just ask him, Dickson, how many viral genome types are there? And he would say 7 over and over. And when you had Dickson on a podcast, you knew it was not going to be boring, right? Except the times when he slept through it. But he usually woke up again. But I loved being on podcasts with him. He was a great raconteur. He was enthusiastic and going to be missed because he was a great addition to all of these podcasts. So he hasn't been here for a while, but you have a great library of videos and podcasts, including the early ones where there's no video; it's just the two of us talking. And I just wanted to make one reminiscent. I remember early on in TWiV, he said norovirus was named after Norwalk, Connecticut. And oh my gosh, did we get emails? No, it's Norwalk, Ohio. And so every time he mentioned norovirus after that, he used to say, from Norwalk, Ohio, not Connecticut. But I think it was the first time he realized the reach we had, that people were going to correct us if we said something wrong.
They're actually listening. Yeah.
They're listening, yeah.
So, Vincent, I remember early on when I visited your place, if I remember correctly, Dickson had an office in your office or something like that. Is that right?
Yeah, so of course for years he had his own office. And then when his lab closed, they moved him to another office. It was actually right above the emergency room of the hospital. And I know this because I helped him move his stuff there. And then they took that away from him. And he said, I don't know what I'm going to do. And I said, well, we have this desk outside. So there was a suite of 3 offices, Saul Silverstein, me, and Steve Goff. And then there was a suite where you put assistants, 2, and 1 was empty. So I said, why don't you sit there? And I had offered him that originally before he went to the space above the ER. And he said, no, I can't be there. And then he came when he had nowhere else to go. And he would come there every day. And it was right next to the door. So whoever came in, he would look at them and decide whether to talk to them. And that's how he started talking to Daniel, because Daniel starts working in Steve Goff's lab and he passes Dickson and Dickson starts talking, because Dickson had taught you years before.
Yeah, well, I don't want to jump out of turn, but because I think Rich, is it your turn? Because I'll jump in.
No,
I'll be
Alan's
next.
I'll jump in. Who's jumping in? Who's
Alan
next?
is next.
I'll be next because I was the next to join the show. We're doing in show.
Yeah, go ahead, Alan. Go ahead.
So I met Dickson in the early 1990s when I went to Columbia for graduate school. He was in the microbiology department. He was the only parasitologist in the department. And that seemed kind of cool. And I got to know him a bit during graduate school. One of my classmates had actually been a technician in his lab before coming to graduate school at Columbia. And by the way, Steve Buck, if you're listening, drop me a line. We lost touch. So I knew a bit about Dickson's work, and he seemed like an interesting fellow. And we interacted at the microbiology department retreat, and I discovered that this was somebody who wasn't just a parasitologist. He actually knew a lot about a whole bunch of different things and was just fun to talk to. And then my wife, who went to Columbia Medical School starting in 2000, and I had finished graduate school by then. She had him as a parasitology instructor. She said, "Did you know Dickson Despommier?" I said, "Oh, yeah. She said, "He is wonderful. We all love him. He's so great." The medical students just absolutely adored him, and he won teaching awards from them. But then I kind of lost track of folks there, got busy with my own work. And then in 2008, autumn of 2008, I looked this up. I went to Mexico City where I was covering a conference for the New York Academy of Sciences. I was one of their in-house science journalists, not in-house exactly. I was a freelancer, but they hired me to do a lot of conference coverage. And it was one of these big ideas conferences. You know, the mayor of Mexico City spoke, a couple of billionaires spoke about addressing the challenges of the 21st century and how Mexico City could, you know, build itself as a hub for innovation and so forth, and cool stuff like that. And I saw on the agenda Dickson Despommier, who was going to talk about vertical farms. Wait a second. This has got to be the same guy, but what's going on here? And I caught up with him at the meeting, and then I heard his talk, and you know, I remembered, yeah, you know, he always was interested in, he had this omnivorous curiosity. And he told the story in that talk. It was actually one of the first major events where he was presenting the idea of the vertical farm. He hadn't done his book yet. He hadn't gone on Colbert. And hearing his talk and talking to him at the meeting, I thought, you know, this actually really fits. This is the kind of thing Dickson would do. He had this highly unusual capacity to dive into a complex, depressing subject. In that case, it was global food security, and he, you know, also climate change. And then if you read The New City, which I would highly recommend if you haven't read any of his books, or even if you have, it really crystallizes a lot of stuff. And it's, you know, he jumped into these topics that most of us read about and are aware of and say, wow, you know, we're screwed. We're changing the climate. Our grandchildren are going to have to deal with this. And it's just, it's a big old mess. And the structures we have to deal with it are not adequate. Dickson could go into something like that and come up brimming with optimism about the future. His approach was, this is a great opportunity. We can build new things. We can fix our cities, you know, and here's how, and we can figure it out. And I think, you know, the vertical farms have gotten some pushback as expected. But I think it was never really about a specific approach or a specific technology. I think he was really about getting people to think about this, you know, these things that we push to the back of our minds or aren't even aware of. You know, where does our food come from? And not only think about it, but to believe that it could be fixed. And that is kind of the memory that I take of him. And also, of course, you know, just the wonderful stories and the delight of just sitting around talking to Dickson.
You know, in that vein, just before he had joined TWiV, he had written his book, West Nile Story, right? So he had shut his lab and he was teaching, and he said, what else am I going to do? And West Nile happened, so he decided to write a little book about it.
Yeah.
And that was our first episode of TWiV, West Nile Story, I remember. And after we recorded it, he said, okay, well, that's all I know about viruses. So I said, that's okay, you're staying. And then after a few episodes, you know, we recorded on Fridays at 2 p.m. back then. And Dickson said, I want to do things on Fridays. So I said, all right, I got to get more people. We'd had, I think we had you on as a guest, right?
Yeah. Yeah, I came on as a guest to talk about science journalism. And actually, when you contacted me and said you'd launch this podcast and what do I think of it? And I listened to the first couple of episodes. And I've told this story many times before. You know, I thought, wow, this is a great idea. But the thing I haven't mentioned, I think, is not only was I impressed with your use of the narrowcasting, you know, podcasting technology, because this is a great thing. The people who would listen to this are scattered around the world, and this is how you'd get to them. But I was also really impressed at the time that you had picked Dickson as your co-host. Because I thought, yeah, that's who you want to have a conversation with about anything. It could be about, you know, whatever. But he was a great starting point for the show.
Oh, it was clear that he would be the one.
Yeah.
His lab was closed, right? So he could put the time in, but also he was a great teacher and a great speaker.
Yeah.
So it was a no-brainer. And this is typical of Dickson. He just said yes, right?
Yes.
Without any idea of what he was getting into, I think.
Yes.
Dickson, we're going to do this every Friday forever, you know. So that was really great. So Alan came on, and then I don't know, we had this desire to expand it, and so I think Rich came on next as a guest, right?
Yeah, you were doing virus families.
Yeah.
Okay, and you came up to pox viruses and said, "Oh, I know a pox guy." So I came on and, like Alan, did a guest thing, and then came back several times, and then it got locked in because it worked. It was good. I know Dickson only through TWiV, but only in quotes. All right, that's a lot. Raconteur is the perfect description. He could talk about anything and knowledgeably. Sometimes not knowledgeably, you know, he'd go off the rails, but that's okay, you know. So the West Nile thing, because that keeps coming to mind. Is that just Dickson flipping out, or does that grow out of his medical ecology thing, among other things? You know?
Maybe it did to a certain extent, right? He knew some of the players in flaviviruses, right? And so maybe that was part of it. But I think he just saw an interesting story, and he's a scientist. He understood it. The book is really a low-level book, right? It's not highly technical, so anybody can read it.
So, you know, boundless curiosity, boundless interest. He surprised me all the time. Okay, you know, you turn around and there was something else about Dickson that you weren't familiar with. You know, tying flies for his fishing. He has a collection of pictures of fish, I think, and flies and stuff that he framed himself. I don't know if he took the pictures himself, or he's a terrific photographer. Okay, he traveled all over the place so that there was pictures from all of his travels. Some other subject would come up. So, he's not gone. I decided that he's going to take over Tinkerbell. Okay? So, for those of you out there, Tinkerbell isn't around today because it's too cloudy. But when Tinkerbell shows up, it's no longer Tinkerbell. It's Dickson.
It's Dickson.
Cool.
Dickson's still with us.
I love it.
The room where he recorded the podcast, right behind him, he had these benches where he had all his flies and he was a framer. He could make frames for pictures. He had all the wood and the saws and everything you needed to make picture frames. And so that he put a lot of his flies in frames. He did. And then at the incubator, I had bought a watercolor, and I bought a cheap frame at some store. And he came in and he said, "No, you can't do this." And he made a frame for it. He took the print home and he made a custom frame, which is gorgeous, right? You know, custom frames are just beautiful, and that's another thing that he could do. Oh, and he also painted, right? I have a bunch of watercolors here and at home. He donated to me because he didn't know what to do with them, which are all on a similar theme, which are beautiful. But he did art shows at Columbia. I think he painted in other media as well. So yeah, did a lot of.
So the New York Times obituary was very good, but it you know, it's only scratches the surface because Dickson. Well, we said it, boundless curiosity. And he did, you know, and he acted on his curiosity and pursued things with passion and with talent. And Dickson, I'm just not going to let you go. Okay?
I think I told someone that I found that obituary to be really, really nice and an incredibly narrow slice.
Yeah.
Yes.
Very nice. Yeah. I mean, it's really all about indoor farming, right?
Yeah.
Yeah.
There's
I mean, they only had a few thousand words, and how are you gonna...
Yes, exactly. Okay, so next to join was Kathy.
So I too was a guest, but I was a guest here at Michigan when Rich and Vincent came, and we did a TWiV from here. So I didn't meet Dickson even online for quite a while, because then I did a couple of episodes when Dickson was fishing and Rich and Alan weren't available. And then when I joined is when I first really met Dickson online. So, and then I met him in person at TWiV 300 in Washington, D.C., and then met him again, or saw him again at 400 in New York City, at 500 in Austin, and 1,000 again in New York City. And then, of course, many, many times in person. And so my description of Dickson, there's a couple of terms. One is that he was a polymath, which is someone who has great learning in several fields of study. So that's definitely the case. We've heard about his knowledge about parasites and, of course, their hosts. And although we share that slice with him here on Microbe TV, then there's the other things, his advocacy for vertical farming, for which I think he probably would fit the term autodidact, and maybe many of his other pursuits, someone who teaches themselves the information, or he goes out and finds the information that he needs for that. And so I think he was really good at that. And then the new city, the use of timber and engineering to help us build sustainable cities. And so he brought his knowledge from these various areas to the TWiV podcast, not as a virologist, but just as somebody who could come in with these sort of orthogonal questions just from somewhere else that were brilliant, that we hadn't thought of. And so that those prompted some really interesting discussions. And so I really appreciate him for that. Then, of course, he had lots of other passions too. His Jazz Picks of the Week series that many of you heard, his great admiration for photography and all the photo contests, particularly like the Nikon Small World one, where he would almost always pick that one and we would look through those photos and ooh and ah. But in part, it was because he was such a good photographer. And Vincent's already told you about his various websites. And then for me personally, sometimes Dickson would send an email to all of us and say, Happy Thanksgiving or something. And people would write back to him, but I would write back to him separately, or we would just have email threads about other things. And so pretty recently, within the last 6 or 8 months, he sent me a whole series. It's a huge series of pictures that he's taken from around the world of things with spirals in them. And so he said, I don't know what to do with them. I can't get somebody to publish them. And I said, well, maybe they could, be a website or something. So it's probably making more work for Vincent. But I think there's over 1,000 photos and they're beautiful. I mean, it's just an eye for that kind of thing. And so, yes, you did have to do some fact checking with Dickson. In fact, the episode where Vincent and I did a thing about what's a normal day for a virologist like. And I started out by saying, well, if it's the season of the year when I'm walking to work, I'll be listening to a podcast and I'll have to try and remember by the time I get to work all the things that I need to look up and see whether Dickson was right about. some facts never quite fit. So I'm pretty sure I caught the norovirus one early on. But the one that he said at least twice, and I had to correct him, and he took it very good-naturedly, was that Harry Ginsberg, the adenovirologist there at Columbia, chair of the department, that he had played football for the University of Michigan. And he did not attend the University of Michigan, let alone play football. He attended Duke. And from looking at Harry Ginsberg's bio on the Columbia website, there's no mention of him playing football. So I
also, from looking at Harry Ginsberg, the notion of him playing football is a little incongruous.
Well, he did actually. He did.
He did.
Football.
Did he?
Oh.
He was a offensive lineman, little guy, very fast, right?
Oh,
yeah.
Oh,
and
well,
did
and
he do
that also would have been back in the day when, before the special teams rule and all that unlimited.
But his knees were all messed up.
Yeah.
And he always used a cane, and I asked him once. He said, "I had my football years."
Yeah. Okay. So was that at Duke that he did that, or at high school?
No, I don't know. Actually, that's a good
Yeah.
question.
Okay.
Sure.
All right. Anyway, back to Dickson. He was really kind, warm, very generous, and I was really fortunate to have met him those various times in person and online. And whenever I would see him, he would so genuinely say, "Kathy, it's great to see you." And it made me feel really special. And I have the feeling he was that way toward many, many of you and many
Yes.
other people. And so I think I've said pretty much everything I wanted to say. Life was really a joy for Dickson. You could tell that he was passionate about things, but he was so just happy about things. I'm really fortunate to have known him, and I will miss him.
All right, now next, Daniel and Brianne are very close. Daniel joined TWiP in 2015, I think, but didn't join TWiV until really the 2020, the pandemic beginning. So Brianne, I think, came to TWiV in 2017. Is that
Mhm.
right? So let's go next with Brianne.
Okay. So as Vincent mentioned, I came to TWiV in 2017, and before that, I was a TWiV listener, and so I already appreciated Dickson's sort of broad knowledge and being a polymath and all of the things that everyone else has phrased so well, and his ability to be a storyteller. But the first time I actually met Dickson in person was once when I came to Vincent's office to see the wall of polio, which was also the first time I was a guest on TWiV. And Dickson, of course, was there in the seat outside of Vincent's office. And so I met him as soon as I came in, and he, gave me a big smile and shook my hand. And he had a lot of samples from his time as a parasitologist. And so almost immediately upon sort of walking in and being introduced, he really wanted to show me a lot of parasites in jars. and sort of had this boundless enthusiasm, but also this desire to sort of share this exciting information with someone else who found it exciting. And the biggest thing I remember, especially that day, was just how warm Dickson was, how we sat next to each other at the table in Vincent's office. And, I was this, random listener who would come by to be a part of it. But as far as he was concerned, we were old friends, and I was as much a part of the conversation and as much a part of that podcast episode as he was. And he was just so excited to be able to include me and talk with me, and always from then on seemed to always share that warmth and enthusiasm. And so I will always think about all of the things I learned from him and all of the ways I think about him as an amazing teacher and ways I try to teach like that, the ways that I want to tell stories like that, his art that is unbelievably wonderful. But to be honest, when I think of Dickson, I think of warmth and joy the most. So as on that day that I went to Vincent's office, Vincent actually took some pictures and put them on Instagram. And as a fangirl that I was, I screenshotted all of that. And I was trying to find them right before this, and I can't find them. I will keep looking. But Vincent took a picture of Dickson and I sitting together recording, or I think maybe right before we were recording. And in the picture, the two of us are sort of looking at each other, talking and laughing, our heads kind of back laughing. And that sort of sharing information with warmth and joy, and that ability to include and desire to include everyone in your passion is really very much Dickson to me. Similarly, I at one point, got in touch with him because one of my closest friends here at Drew works in our environmental studies program, and I thought about his work with vertical farms and how that might be of interest. And so she started having him come to classes, and he was sort of like, of course I'm coming to Drew. Of course I'm going to come do this. How can I be part of this class? And he didn't say no. He always said yes and jumped in to have all these experiences. I really appreciate that. and I wasn't obviously there for this part, but I think about it, that I really appreciate him saying yes to Vincent when Vincent sort of started TWiV, because I can imagine if, he was a pretty big deal in Paris as a parasitologist. He was rather well established as a parasitologist. And Vincent said to him he wanted to start a podcast about virology. And I could imagine if someone came to me and asked to start a podcast about something that was very much not my field of interest, I don't know that I would necessarily jump at that and have that courage and that delight and optimism to find out more and learn more, just like Kathy was saying about being an autodictat. And so I also think we should, give some big props to Dickson for jumping in so readily in the way that he did. And I, like everyone else, am really going to miss him because his optimism and inclusion and positivity always sort of, especially when I was first coming to TWiV, really helped me kind of feel part of the group and feel confident. And I think that's generally part of Dickson in terms of whatever group he's in, trying to include people and make them feel like a part of it. And so
He would never
appreciate
judge
that.
you. He would
No.
never say, "Oh, you're young and you don't know anything." But no, you're just an equal member, right?
That's
the way he was.
exactly right. And that was so helpful to me.
Yeah. So I wanted to mention that at some point he had to leave this outer office because someone else needed it. It wasn't too long ago, actually. And he had, as Brianne said, put all these parasites there. So I had to bring them in my lab and do something with them. But then he had hundreds and hundreds of books, more than you see behind Daniel. And I had to sort through them. And half of them are here at the incubator. In fact, I put in a bookshelf and it only has his books on it. My books are just on this little shelf behind me, but in the other room. But they're not just on parasites. They're on insects and volcanoes and oceanography, every subject you can imagine, right? He's got books on them because that's what he was interested in. All right, so next comes Daniel.
Hello. So I first met Dickson back in the early 1990s. He was my parasitology professor when I was a medical student. And 20 years later, it's summer of 2013, and I start at Columbia, and I'm doing research in Stephen Goff's lab. And, that anteroom, I come to and fro, and there's Dickson. And so Dickson, we start talking, and quickly it occurs to me, I know this guy. And so I was like, I know you. And to Dickson's delight, I said, you know what, Dickson Despommier, 20 years ago, you taught me parasitology. Not only do I remember you, but I can tell you not only what you taught me, but what you had for lunch before the first lecture, which was sushi. So he's just such a memorable guy. 20 years later, not only do I remember what he taught me, but I remember what he had for lunch. And, and we became friends, with that reconnect. And we did a lot of stuff. We went fly fishing together. We shared that passion. We went to a lot of events. Before I think everything else, he and I were friends. But then as we talked together, that became Parasites Without Borders, right? We started talking about, the textbook that I used, which had been sort of sitting dormant since, probably the edition he taught me from. And then we talked about creating another version. And then, hey, when we create this version, we don't want to charge people. We want to get it to the people and places that need it the most. Let's create a way, instead of Elsevier charging $100 a copy, that we can give it to all these people throughout the world. So we started that. And I guess it was, about 10 years ago, 2015, that I joined This Week in Parasitism. And I remember those first episodes in recording in Vincent's office. And the biggest thing was to have Dickson actually keep his face towards the mic because, he turns all, wants to look at you, the whole format. I think he did better with the screen, with the Zoom, because he keeps looking straight ahead. But yeah, I think everything that people have pointed out, and I think that all of our listeners, right, should feel the same. He loved teaching, and I felt like that New York Times article didn't capture really what I think Dickson took the most pride in, and it was teaching. It was sharing stories. They talked about the vertical farming and poo-pooed it a little, but I want to point out college campuses across the country, hydroponics and indoor growing, it's going strong. Stephen Colbert pointed that out. I think we know where the ideas came from. But no, he loved that. And I think everyone, if he met someone and we would go to these American Society of Tropical Medicine and Hygiene meetings, and people would come up and say, Dickson Despommier, I listened to TWiV. Oh, and he would sit down and just regale them. I mean, they were, so I think all of our listeners, they can do the same thing we're doing and say, when did you first meet Dickson? Because he considered all our listeners his friends too. And one of the great things I think about Dickson is he didn't always let the truth stand in the way of a good story. I know Kathy pointed that out.
A long-standing Southern habit, I'll have you know.
My grandmother actually once told me that when I tried to correct her on a point of fact, she's like, "Oh, Daniel, don't let the facts stand in the way of a good story."
A lot of my kinfolk are like this. I'm very accustomed to it. I felt right at home.
But he always had a good story, and I think his optimism. We need to remember that more now than ever. And on the last TWiV clinical update, I talked. Well, I guess it hasn't dropped yet, so it'll drop on Saturday. You can listen to his. In one of his medical ecology classes, he asked 2 questions, and I'm going to add a third, which is what he would always ask. The first was he'd say, "Okay, imagine the future. What do you think the world is going to be like in 2050?" And they'd talk for a bit, and then he'd say, "What would you like the world to be like in 2050? And that would get people more excited. And then he would always follow up, and this is really what Dickson was about. What can you do to make it like that in 2015? This call to action. And that was the great thing about Dickson. So even to his, I can say to his dying day, he was active, he was enthusiastic, he was contributing.
Yeah, he called me a few weeks ago, and he said, I can't podcast anymore. I can't get to the chair effectively and sit down. It's too much trouble. So he said, I'm sorry, I can't do that. He said, I'll try, but of course he didn't. He wasn't able to do that. I remember when you first came, Daniel, he talked to you for a while, and then he came into my office one day and said, Vincent, this guy, Daniel Griffin, he's a MD. He knows about parasite infections. I think we should bring him on TWiV, and he could do clinical cases. I said, yeah, it sounds good to me. Let's do it. Because we had finished 20 episodes of basically individual parasites. And then what are we going to do? Start talking about papers. And I didn't feel that Dickson had the heart for that. So that was great that we were able to do that. Yeah. All right. Who was next, Angela or Jolene?
I'm
I
pretty
was
sure
2021.
Angela started before me. Yeah.
Okay, Angela.
Yeah, so I joined TWiV in 2021, but I didn't actually meet Dickson until 2023, April, at TWiV 1000. That was the first time we met in person. And I think that, we talk a lot about infectious agents on our podcast, and I think that Dickson himself, he had such an infectious personality. He was, since the first moment that I met him virtually, like Brianne said before, so welcoming, so jovial and cheerful. When there was an episode where Dickson was going to be on, I was even more excited because I was like, Dickson's going to be there. He's always going to be smiling. He's always going to have hilarious things to say, little anecdotes, and also very smart. Everybody has already highlighted how his breadth of knowledge in so many different fields. But I think that what I'm the most marked with from meeting Dickson, which obviously was not as much as everybody else here, but I think that he had such a beautiful soul, and just speaking to him, he was such a wonderful human. And I think that I'm so happy that I had the pleasure of meeting him in person, even though it was only once. And I remember when I started on the podcast a few months in my own research, we were looking at T cells and parasitic infections, and I emailed Dickson and I was like, Dickson, can we do a Zoom call? Maybe we can discuss it for a few minutes, just you can help me answer some questions. And we had like a 2.5-hour Zoom call talking about trichinella and every other parasite. And he was so generous with his time. And I think he'll truly be missed. And I think that he was, one of the words to describe Dickson was infectious, welcoming, jovial, which I think are the most beautiful things that people can say about you when you pass. Like for people to remember how good you made them feel, which, like I said, he just always made me feel so warm, so welcoming. And yeah, and also I have, for those of you that are looking at the video, I have this selfie from TWiV 1000, which is so beautiful. And I think that, that I'm so happy to have actually met him in person and that he will definitely be missed.
So you just met him once at 1000, right?
Only once. Yeah.
I remember you guys talking quite a bit.
We did. I have multiple selfies, actually. I just one of them. I was like, Dickson, this might only be the first time. I need to take a picture. And he's like, oh, yeah. Let's do it. He was so sweet and so tall. I never thought he would be that tall. I remember I saw him and I was like, Dickson, you're so tall. He's like, yeah, so are you. We talked about that for like 15 minutes.
And we're not. So I remember, for some reason, this jogged my memory. Early on one of the podcasts, one of the TWiVs, I think, before we had video, it was all about, I think, viruses getting into cells. And he said, safe crackers, all of them. It was so perfect.
Yes.
And always, we used it in a title later, safe crackers. It was just perfect. Loved it. Could come up with things like that. Okay, Jolene.
Yes. So I don't have a lot of different things to add because Dickson was just such a wonderful storyteller. I was captivated by listening to the very first episode when I was a graduate student, and I was hooked from then on, from episode 1 forward. And I just really appreciate his fisherman style of telling stories. The fish just got bigger. Just did. I only had the pleasure of recording with him in person, online a few times. I met him at TWiV 500. Which was lovely. And I just want to add that he really took correction very well.
Yes.
He was humble and graceful about it. And I strive to be like that when I mix up my things about the 7 genomes of viruses. And I wrote a little Tonka for D-Cubed that I'd like to read. So it goes, scientist relates borderless intrigue taught through captivating tales, natural relationships, fascinating friends recall.
Cool.
Oh.
And I just want to mention, I put up a short blog post about this as well on my Alan Dove site, not as a self-promotional thing. I just wanted to mention I went to Wikipedia to see if they had a picture of him, Vincent, and I found the picture and I saw the photo credit. I said, oh, well, isn't this nice? So I posted that on my site. It's a CC license, so I'm allowed to do that.
Yeah, totally. The picture is funny. He couldn't see and he got a pair of glasses. So the photo is taken with these glasses, which he never wore. He wore the glasses for 2 weeks and then he showed up without them. He said, turns out I just had a transient thing and I don't need them. But that one picture, which is on Wikipedia, has the glasses on, which is not him at all, right? It's just so funny. All the things that you remember.
Yeah,
Okay.
but it also has a smile on his face, and that is definitely him.
Oh, he always smiled, yeah. I don't remember any time. And he came here a few times. I don't remember why he would come here, Dan. Was it to do TWiP, Daniel?
Yeah, I think he, even sort of near the end, he would always show up to do TWiP. We'd usually have lunch before, because he was not a huge fan of reading the papers himself. He'd always want me to read them. And so we'd go through it. So.
Well, he also
Yeah.
he often liked to talk about going to lunch as well. I know that he mentioned a number of restaurants to me, and every time would then follow it up with, "Oh, well, I'll have to take you there."
Yeah, there was a nearby Irish place where I think that was where a lot of the early PWB meetings were. Coogan's, if people are familiar.
Yes, so I know
Oh,
Coogan's
up in
well.
Columbia,
Yeah.
But down here, he used to come to record periodically. And then it would be late. And he said, "Let's go get something to eat." I said, "Dickson, it's 8 o'clock. I got to go." So I put him in an Uber to home, and yeah, he did that quite a bit. That's right. But he always said, "This is great. What you did here. Next year, it's going to be twice as big." I said, "Why do I need it to be twice?" Because you're going to get more people, and you're doing great. It was always enthusiastic like that.
Yeah.
He really liked it here. Yeah. All right.
He lives on in many, many hearts and minds.
Indeed.
And so for his sake, I will try to imagine a healthier world with greener cities and streams full of trout.
All right, well, I think I'm going to leave you guys, right?
Thank you,
So
Dick.
you can
Thank
do
you,
your
Daniel.
snippets and stuff. But hey, it's great
Thank
to
you,
see
Daniel.
everyone.
Good to see you,
Great to
Daniel.
see you.
I'll
Great
see you
to
in
see
a.
Bye.
I'll see you in a bit, Daniel.
Yes, I see a lot of you, Vincent. So we'll see you in an hour.
Bye. All right. We got some news here. I think Alan, you put.
Let's see, what did I put in here? Oh, yeah. So the first two I put in. First one is an MMWR, which is now publishing again, report. It's a notes from the field. They did a seroprevalence survey of H5N1 flu in cattle. This is the dairy cattle bird flu thing we've been talking about for a while. And now we finally have some data on seroprevalence in veterinarians who work with cattle. Because that was a sample, kind of, as you might expect, dairy farms are not keen to have their workers sampled for this because it's not something they really want to talk about. But they got samples from 150 bovine veterinary practitioners. Those are vets who practice on bovines, not bovines who are vets, just to be clear. And 3 of them had positive antibodies for, suggesting recent infection with avian flu. That included 2 of them who didn't have known exposures to herds that were known to have this virus, one of whom didn't even practice in a state known to have this virus in the cattle, which suggests, somewhat unsurprisingly, that this virus is probably a lot more widespread than we realize and is spilling over repeatedly into people who have contact with cattle. Whether it gets any further than that, remains to be seen. And I've also linked to an NPR story that summarizes this.
Let me just add before you go to the next one. Yesterday on Clinical Update, Daniel gave a story about some cat infection. So a number of these individuals who work in the dairy farms, they have cats at home, right? And so in a couple of, I think 3, the cats got infected with the virus from the cow via the cow. Now, in people, this is a pretty mild infection. It's conjunctivitis. But one of the cats died, and they had a pretty severe illness. A couple of them recovered. So it's being transmitted to cats and other animals, too, from these workers. But what's interesting is to a T, all of the workers would not submit to H5N1 testing because they said, "I don't want to lose my job."
Yep.
That's
So,
sad, though.
Well, why would they lose their jobs? They just might have to stop working for a little bit, right?
Because the farm would be outed, I guess. I don't know.
Yeah.
Yeah, their employer would not be happy about that. And people who work in agriculture generally, and, these dairy workers are going to be in this category, tend to not be exactly well paid enough that a few days off work would be, they don't get a lot of benefits like paid sick leave. So that could be a financial catastrophe for them. Even if they decide to.
One of these workers transported unpasteurized milk from the farm to somewhere else, maybe to the pasteurization plant, because I don't think they do it on the farms, right?
Depends.
But,
If it's a big
that
farm, they
depends.
do. But yeah, depends.
That's how he presumably got infected. Big tins of unpasteurized milk.
Have we, I have not been on an episode that did this. Have you guys gone over the genetics of these viruses that, a couple of different outbreaks in cattle and the bird viruses? Is there, I'm interested in whether the genetic variation makes any sense or tracks with these different outbreaks and stuff,
So-
and how it might relate to what we know about transmissibility and
Yeah.
et
So
cetera.
I don't know if we did it, but what happened was there was a spillover from birds into a cow. I forgot, Daniel mentioned the state, and all the other cow infections derive from that
I believe
single.
it was Texas.
Texas? And all the other cow infections derive from that spillover. And there were changes, right? I think it might have been a reassortant event. So we should probably
Yeah,
get
I
someone
would.
on to talk
I'm
about
really
that.
interested in that,
Yeah.
to put that together.
And then, of course, there's the avian lineage, which is separate,
Right.
right? So two different. Yeah, it's quite. All right. Sorry, Alan.
Sure, no problem. That's the point of bringing in the news stories is so we can have some discussions. And then the other thing I picked up is an article in Science about Elizabeth Bick, she's a microbiologist, but she's also been at the forefront of the science integrity efforts to, find things like duplicated figures in papers, and people have manipulated it, or sometimes by accident, a lot of times, unfortunately, not so much by accident, and has done a lot of data sleuthing, as they say in the article. She won an award for that, a $200,000 award for her work in scientific integrity, and she has donated that money to help fund other people's investigations of scientific data. I thought
Good
that
for
was
her.
worth a mention. So
Yeah.
good job. Thank you.
She's very persistent about
She
calling
is.
out the journals
Yes.
who refuse to take down data that have been reported.
Yes.
I have not seen her since I stopped doing much Twitter.
Yep.
But her Twitter was amazing.
She is on Blue
Sort of-
Sky.
Okay, I'll have to look and make sure I can find her, because the sort of level of persistence and level of detail in demonstrating the issues with some of the data was amazing.
Yeah.
That's
Yeah,
how
it wasn't.
I know her.
It wasn't
from
just
Twitter.
like some people say, these figures look an awful lot alike. It's like, the prosecution will now present its exhibits.
Mhm.
Yeah.
I wonder
I'm
how
looking for
many
her
of
on
those
Blue Sky.
were Western blots. I remember in
Yeah.
almost all the retracted papers that I've seen are all mostly Western blots. I'd
Yeah.
say
She
like
is not
90%.
somebody who goes off half-cocked, though. So, yeah.
Yeah. I'm gonna follow her on Blue Sky as well.
I just followed her on Blue Sky, so now I can see some of these images again.
Yeah.
So, Jolene, did you put the Texas story in?
I did, yes. There has been a developing measles outbreak in Texas. There were a few cases in Houston, but this is actually over in West Texas on the border with New Mexico. And the Texas Department of State Health Services puts out an update every Tuesday and Friday. So as of today's update in Texas on our side, there are 90 cases that have been identified. The majority of them are in unvaccinated individuals. There are 5 vaccinated individuals that have developed measles symptoms. And so far, there are 16 individuals who are in the hospital. The majority of these are under 18 years old, most of them kindergartners or below. So this is going to continue to spread because measles is very contagious, and the levels of vaccination are incredibly low in these communities over there where the outbreak is centered.
So I learned that in Texas, overall, the measles vaccination rate is 97%, which is quite good. But it's in these isolated counties where you have the medical exemptions, which go to a ridiculous level. And that's a
So
big
that's
problem.
almost 20% of the cases are hospitalized.
Yep.
That's right. Yeah.
It's a lot, yeah. That's quite a bit.
But there is an effective vaccine.
Yes.
Yay!
Highly
vaccine.
effective vaccine.
2
I don't
doses.
understand why parents are not vaccinating these kids. They're influenced by anti-vaxxers, but I don't know why.
They're
they would do
also some of them it's a religious issue, which is, I'm not saying that's necessarily better, but it's not all anti-vax.
Yeah, well,
People drill down a YouTube rabbit hole. Some of them have a religious belief that just is against vaccination, and this is the first infection that's going to pop up when you do that.
but also Texas has the highest rate of medical exemptions in the country.
They make it very easy, I think, to
it's very
get the
easy.
medical exemption.
It's not right. You should not be more than 1%. Remember, Rich, we did that vaccine thing in Irvine years ago, and the guy said medical exemption should not exceed 1% in the U.S. And there's these counties in Texas where it's 30, 40%. So it's a big
I'm just
problem.
looking at the map because they break this down. They break the cases down by county. And not surprisingly, the majority of the cases are in counties that are right next to each other.
Mm-hmm.
So I learned from Daniel last night that there are 300,000 children in the U.S. who are unvaccinated against measles. Now, most of them are scattered around, so that's good. But as you see here, when they cluster, it's a problem. Okay. Let's do a snippet. Dixon would want us to do some science,
Yes.
right?
Mm-hmm.
Yeah, I think he would be fascinated by this. So,
This article has, I read it, what's today, Friday? I
Yep.
read it Wednesday, and it's been haunting me ever since. This is so cool.
I skimmed through it when Jolene sent the link out over email, or sent the paper out over email. I saw the title and I was, wait, and then I read the abstract and I said, I have got to get back to this. This is going to be good.
Yeah, and I specifically thought, I was like, oh, I really can't wait to hear what Julene has to say about
Yeah,
this.
I got questions, Julie.
Yeah.
So, Kathy, can you give us a summary?
Well, sure. Everybody's probably curious about the title. The title is Experimental Validation That Human Microbiome Phages Use Alternative Genetic Coding. And I didn't prepare something about the authors, but the first author is Samantha Peters, and the last author is Robert Hedich.
I can do affiliations if you want.
Yeah, and I don't know if they're if there's more than one.
Yeah,
first
it's
author.
there are no levels here. The group is from Oak Ridge National Laboratory in Oak Ridge, Tennessee, University of Tennessee, Knoxville, University of California, Berkeley, University of Pittsburgh, School of Medicine, Lawrence Berkeley National Lab in Berkeley, and the Chan Zuckerberg Biohub in San Francisco.
So the title, I think it threw me a little bit because I just put big parentheses around part of it and said, we can omit this. The experimental validation that human microbiome phages use alternative genetic coding, I think would be enough. And the point is that this had been something that had been predicted from bioinformatics. And now they've gotten some really cool data about it. So the short version of the summary is that the genetic code that you learned about in high school that has 64 codons, one is a start codon and 3 are stop codons. That's not always how translation works. And these authors are going to show you that one of the cases when it doesn't work that way is these bacteriophages, which are viruses that infect bacteria, and in this case of the human gut microbiome. So the slightly longer version of the summary is that you may remember that there's 4 nucleotide bases, and the order in which they are found in a messenger RNA helps define what proteins can be made from it. These nucleotides are read in groups of 3 called codons, and then they're read by decoder molecules, which are called tRNAs, that can base pair with that 3 nucleotide sequence. And at the other end of the tRNA decoder molecule, there's generally an amino acid. And because of that, that's the amino acid that it puts in. Now, when you get to the 3 so-called stop codons, it's a little bit different. And there's not tRNAs that will put in that are going to read those codons in general. And Vincent, you put in this somebody put in this cool YouTube link. I hope you'll put that in for the listeners if they want to review how
that
protein
was
translation
me. I
works.
was looking for
Okay.
something that would show how all that works.
Right. Okay. So Rich has put that in there.
And
for
you need the codon table that somebody else put in as well to help out with that.
Right. So the way we got to this 64 is that, of course, there's these 3 positions and there's 4 nucleotide possibilities. So you have 4 times 4 times 4. So the 3 stop codons, which in DNA sequence read as TGA, TAG, and TAA, and this paper tends to do everything in terms of the DNA codons. So maybe that's how we'll just talk about it. But if you start with the start codon, which is ATG in DNA or AUG, and you find that sequence in your string of nucleotide letters of a nucleic acid sequence. If you start with that ATG, then you can read in groups of 3 until you get to a stop codon. But there are mechanisms, first of all, where ATG is not the only thing that can be used for a start codon. So there's exceptions to that rule, but there are exceptions to this rule of these stop codons encoding stopping of protein synthesis. And that's because there are tRNAs that somehow can have an amino acid attached to them and recognize one of these stop codons. And so that's been predicted to be the case for these phages. And the investigators go through and do that in this paper. And there's 2 aspects of that I wanted to point out. First of all, they use some really cool centrifugation and enrichment techniques because these phage proteins are in very small amounts. And that this finding provides sort of a temporal regulation of when certain proteins are made for the phages. And it's kind of a just-in-time thing. It is the hypothesis that they seem to be made with this alternative codon usage at late times after infection when you need to make certain capsid proteins and so forth. So I think that's a cool aspect of it. Take it away, Jolene.
Awesome. Thanks, Kathy.
So I have a question for people, because this comes up first in the abstract, though it comes up elsewhere in the paper, where they talk about alternative genetic codes, and they spend a lot of time talking about the fact that this that they're seeing the use of code 15 here. And I didn't know. So, I mean, I kind of knew there were alternates.
Yeah.
The Wikipedia, and they talk about 11 a lot here, and there's 37 different ones, and there's a whole table. I had no idea until I read this.
So, the so-called universal genetic code that you learn about in all of your biology classes is not actually universal, but it's mostly universal. And in some organisms, they'll use 1 or 2 or maybe
It's
up
more
to like
guidelines.
8 or 10.
Exactly. Yep.
The majority of the changes are in what we call the start, and so they can use different codons to start, and then they can use different codons to stop, or they can, instead of doing a stop in the protein translation process, they can add in an amino acid instead. And so I didn't realize there were 37. I thought there were 33 because I used the NCBI standard, which has all 33 of them listed.
Well, I will admit, though, if you look at this Wikipedia page, some of them are listed as none.
Oh,
so
okay.
I don't entirely understand it, but it's weird. But yeah.
So the genetic code we use for bacteria is number 11. Super familiar with that one. Any time that you're going to be looking for the genes that a virus is making, you're generally going to assume it uses the same genetic code that the host uses. So for all the phage genes that we annotate, we use bacterial code 11. But there had been a paper previously put out by the second author on this one, Adair Borges, had earlier in the same year or the previous year put out a paper that showed a huge bioinformatic analysis of bacteriophages, which had never had experimental demonstration of alternative code use in the microbiome. And they saw that there was an enormous group of phage in human guts and in other guts that looked like they had these alternative codons in some of their genes, specific kinds of genes, the late genes. The late genes are the ones that come on after the virus has gotten into the cell. It has started to take over and it's making more proteins that are going to be used to build virions. And those ones that build virions and the ones that get the virus or the phage out of the cell are the late genes. So they had this big analysis, but what they did in this paper is they took samples from humans. They wanted to show that this was happening so-called in the wild. And they had an adult and a baby, and those 2 individuals were known to have high levels of that one kind of phage that was really shown in the previous paper to have high levels of predicted alternative coding. That class of phages are called crasphages, which are they're just really cool. They're beautiful, but also they are extremely abundant in the human gut. It turns out that most virome samples of human guts, about up to 95% of the reads, can be dominated by these cras-like phages. So these two individuals were known to continuously shed these crasphages. So they took samples from those two people, and they did a really clever thing. First, they extracted the proteins because they wanted to see if it's using the standard code 11, then the protein should be one way. And if it's using standard code or non-standard code number 15, the protein should have a different sequence. So they wanted to pull the proteins, not the genetic sequence, from these samples and actually look at what the protein sequence was. So the first thing they show us is that they expected to find several proteins for each of the two cras-like phages that were found in the two individuals. And it's interesting that they're clustered, but that makes sense because phages often cluster their genes based on function. And in their initial attempt to find phage proteins in these human stool samples failed. Turns out that there's just a lot of human proteins in human stool sample, and that was the majority. So they had to employ an enrichment process. First, they enriched by centrifuging and sequential steps of filtration before they processed the proteins. And that allowed them to get the proteins that would be in the virion, because these late genes are important for building the virion. So you'd expect them to be in the virion. They also later did a validation where they looked inside of cells, and they found some more evidence for these proteins inside of infected cells as well.
Infected bacterial cells, we should
Correct.
say.
Correct.
By the way, I like the way they say phages contribute a relatively small proportion of proteinaceous biomass in fecal samples.
Yes.
I like that.
Yes.
And the crasphages are named after the software that was used to discover them, I think, or something like that. It's not that they're coarse; they're crass for other reasons.
It's cross-assembly. It's because the way that they were discovered was also bioinformatically. That they were so abundant in all these sequence reads, they mixed together read data sets from different samples, and they assembled a genome of a cras-like phage.
Right.
I assume these are double-stranded DNA phages.
That's correct.
And
short tails. They're podophage.
Thank you.
Mm-hmm. Yeah. So on their first pass, or their second pass, I guess, or who knows how many passes, but the successful pass we see in the paper is that they did the preparation of the proteins, and then they trypsinized them. So they treated them with a protease to break them up into smaller fragments, and then they did mass spec. They did two kinds of mass spec. The first kind, LCMSMS, allows them to get a mass spectrum, and then they match those mass spectra to the predicted database of proteins that would be made from the phage genomes. So they predicted the potential proteins translated using the standard code 11 and all the possible proteins for code 15, and probably also any other code that was available. But they would match the spectra with what you should get for each of the codes, and they found 14 or 16 proteins, 14 from one genome and 16 from the other, that matched code 15. So the rest of them were code 11, and these 14 or 16 proteins matched to code 15. And then they show a map saying not only did they match the proteins that should be produced with code 15, but they matched the very junction where the codon is. So you can see that it is the suppression of a stop codon. So Kathy mentioned that there are some tRNAs that instead of, they have an amino acid and they recognize the stop codon. So these will put in the amino acid that you would expect. And the phage actually encodes this suppressor tRNA. So you can sort of, it's all coming together. You've got a suppressor tRNA. You've got genes that look like they would be longer. So I didn't mention that part, but when you first have a genome sequence and predict where the genes are, you use the standard code and you look for starts and stops, and then you think everything in between is going to be formed into a protein. Phages have a very high coding density, typically up to like 95% coding density, because their genes are even overlapping. But when they use the standard code on these genomes, they get a lower coding density, and there are no longer genes in that segment. But when they use the alternate code to predict the genes, they get nice long genes, and the coding density goes up to like 10% higher. So coding density is increased. You have suppressor tRNA gene, and now we have these peptides that come from actual samples in the wild with matches to proteins that would be made using code 15. And they show this for multiple of those 14 to 16 proteins that were matches, they are in late genes, including the tail fiber and other genes like that. They used a second kind of mass spec, which I think is really, really interesting. I'm not going to pretend that I entirely understand how it works, but when they have fragments of the peptides and they run them through the mass spectrometer, and those can generate spectra. They can also say, I see this peak of this peptide and I want to analyze it further. So they pick that peak and they then break it up to generate additional ions that can help them kind of de novo, they call it, basically sequence the amino acids that are in that peptide fragment. And they can get a very characteristic second spectra that will match what you would have based on the amino acid that's present. So
I know you said you're not going to pretend you know very much about it, but do they, at that point, they like pull that peak out and then separately digest it up? And is that when they then add the tag that they later talk about in the paper, that the tags added during the de novo step or something?
Yeah, I don't understand the tag part, honestly.
Okay, good. That lost me. Okay.
I believe
mass spec technology, just as a little side note here, starting in the 2010s, it went through this extraordinary revolution in techniques that, this used to be something that hardcore biochemists would use to identify peptides, and it has turned into almost like a magic box. And there are so many different variations. There's tandem mass spec. Here they're using LC-MS, and you can now drill down into the proteins and do this at scale, to do it proteomically. And I got to the same point too. I was like, okay, mass spec, just wave the wand. Yeah, here's the answer. Which,
let me just try one thing to maybe clarify this a little bit. I'm trying this out. Tell me if I got it right. But the critical element here is that it's called mass spec because what you're doing is you're taking a mixture of peptides that you have generated from proteins, and you're identifying the mass of individual peptides. Okay.
It's a very sensitive, very fast scale.
Yeah. And one of the things that blows my mind is that if you take like a whole proteome, all of the proteins from something, and do a triptych digest and get all of these peptides, they're all, they're complicated enough. So they're all going to have different masses. Mm-hmm. So if you have a predicted sequence and you can, and a predicted digest, and you have these masses come out, you can compare the masses that you get from the machine with the predictions and say, oh, that's this peptide here, which identifies that as being a protein. And what comes out, the thing that blows my mind in this particular one is this figure 2, they show one of these proteins, and they prove from their mass spec that a lot of these peptides have this stop codon in the middle of it, translated as a glutamine?
Glutamine. Yeah.
Okay. So that's proof right there that since you make a peptide that's been translated through a stop codon, that's proof right there that they're using this alternate code. Unbelievable.
Yeah, it's pretty amazing. I think the thing about mass spec that you have to keep in mind is that not every peptide is perfectly unique, right? Some of them are going to be present in different proteins, but that's why the coverage of many different peptides that match to a protein is important.
Right.
So they can do some very I don't even know if it's sophisticated, but you can calculate what you would expect to like randomly get to match, and that's not really your protein. But here they're showing very clearly that they have quite a few peptides that came out matching the proteins that were translated with code 15.
Isn't another key component of mass spectrometry the charge itself? Because it's like the mass to charge, because they ionize the compounds to then... And then I think they ionize them, and then using an electromagnetic field, depending on their mass, then they have the readout. So it's not only mass itself, it's also charge, which will be different in different proteins. I obviously don't understand this too, but this is like what I trying to explain.
That's really what they're plotting in the in the third figure. They have the mass to charge ratio for each position as they ionize it, and that releases these ions that they can then plot.
Exactly.
Yeah. So that is the experimental data that they took samples from humans, and they showed that the proteins that are present in infected bacterial cells and the enriched fractions with phages have proteins that were translated with code 15. And they also have proteins that are translated with code 11. So these phage use more than one code. And like Kathy said, maybe it's a way to regulate their genes so that the ones that are needed come on later. It's probably partly a little bit of defense, right? Hosts turn on and off and cut up tRNAs. And so now they for sure have the one they need because they encoded it themselves. But I think a really other important thing is that this allows us to have confidence in our bioinformatics, that when we see a low coding density in a genome, using another code to translate it might give us a better representation of what's actually in the genome. And that's how we're going to discover new proteins with new functions, is by actually knowing that they're there.
Yeah.
To me, this is it's an alternative way at the level of translation to regulate in a clustered fashion expression of genes from a phage. What we're used to is this being done at the level of transcription, where a phage infects a cell and you get transcription of a certain portion from the host RNA polymerase, and then there will be made a protein that's either a new RNA polymerase or a factor that modifies the existing RNA polymerase, and that lets the RNA polymerase transcribe another section. So you get a cluster of genes early and then a cluster of genes after you make that factor, and there may even be others down the road. This is the same thing essentially at the level of translation, where early during infection, you can make only the proteins that use code 11, okay? And the ones that use code 15, you would, just get a bunch of silly peptides or something like that because there's all these terminations. But then you make a tRNA that now allows you to suppress that termination codon, and you get another whole suite of genes. It's brilliant. They say, they have this sentence: this supports the current hypothesis that alternatively coded phages employ stop codon recoding to prevent premature expression of the structural and lytic phage genes at inappropriate times during the phage infection cycle. So you get done with the early phase, and then you accumulate enough of this tRNA. By then, you say, okay, we're ready to wrap this up and bust out of here. And you can now suppress the codons and make the late stuff. It's just absolutely brilliant. And I thought of Dickson the whole time. He would say, safe crackers.
Yeah,
they're safe crackers.
And a neat aspect of this, regulating at the level of transcription, is you can bank up all this RNA that's floating around the cell that's just, it's not making anything that's going to blow up the cell while you're still replicating. And then when you've got enough of the tRNA and you pass that critical threshold, bang, all of a sudden everything's being translated into capsids and lysis proteins, and you've got all the equipment you need to escape the cell.
So Jolene, I'm doing a paper for TWiV where they of the 3 stop codons, they make them all use 1 stop codon, then they can use the other 2 for inserting unusual amino acids, right?
Yep.
They had to get rid of one of the release factors because it kept recognizing
Oh,
the
really?
stop code. So there are 2 release factors in bacteria
Yeah.
that recognize different stop codons. So in suppression in general, like here, you're putting a glutamine in for a stop codon. How do you prevent the release factor from hitting it, from recognizing the stop codon first and not allowing the tRNA to
You
put in?
don't. You don't. You don't.
So it'd be stochastic, basically.
It's
So
random,
it's going to depend on the concentrations of those 2 proteins. That actually occurred to me. I was like, I wonder if the phage has something to like get rid of the release factor.
Well, there was something. There was I was going to bring this up. There was a sentence somewhere in the paper. I don't know if I'd have to read it again, where at least implied was that in some system, maybe not this, there were phage proteins that counteracted the release factors.
Mm-hmm.
I was going to ask about that. You're not aware of that?
I am not,
Okay.
but that may be known.
But I mean, Jolene's explanation makes sense too. It may just be a numbers
Stochastic,
problem.
yeah.
Yeah.
Thank you, Jolene.
Mm-hmm.
All right. So what should I do? I have all these letters to Dickson. We can't read them, but should I just publish them
Yeah.
in the episode
Well,
letters?
certainly
Yeah?
they should be published. Every now and then when we're missing Dickson, we could read 1 or 2.
Yeah.
Yeah, they're very nice from a bunch of people.
Yeah, really nice.
Here. Okay, then let's go to some picks of the week. Looks like Angela, you're at the top of the list.
Well, I have 2 picks. One of them is just Dickson's tribute. Dickson's obituary also, like by, what is his name? The author was Trip Gabriel in the New York Times, which we've spoken a lot about today, but just like to add it to the show notes if you hadn't already, because I thought that was nice. But also very brief, beautiful, but brief. And then also I added, because I thought Dickson would love this, because Dickson loved everything, literally everything. But this I thought that he would find very interesting. So there was an anglerfish, so a humpback anglerfish, also known as Diablo Negro, which means like black devil in Spanish. Melanocetus johnsii, spotted for the first time ever alive off the coast of Tenerife, which is actually one of the Canary Islands next to where I used to live. So I used to live in Gran Canaria. Tenerife is right next to it in the Atlantic Ocean. And they saw this anglerfish for the first time, and it's ugly. So these anglerfish
And that's a
normally live.
relative thing.
Not if
It's
you're another
true.
anglerfish.
It's
Yeah.
true. I mean, it's not. I wouldn't say it's cute. So I guess it's whatever you want it to be. But I just thought it was very cool that they had never actually imaged one alive before. They've only ever washed up dead. So it was like actually swimming, and somebody took a video of it. David Jara Boguna was the photographer of the anglerfish, and I thought Dickson would love that. And I'm sure he would have anecdotes about anglerfish and more things than I know. But so these fish normally live very, very deep in the ocean, up to 2,000 meters deep. And it's very rare to have it that.
So it doesn't matter how they look. It's dark.
Well, exactly. And some of them are even transparent. Some of
This
them are
thing
even
is
transparent.
terrifying. Okay.
Yeah.
It's terrifying.
I'm sure for
And
it to be that high, it must have been, or that shallow, it must have been sick or something, because it doesn't make sense for it to be that shallow. But it was cool to see it alive.
How
And,
big are
since-
they?
Since Dickson might have provided this information otherwise, a cool thing about anglerfish is, I don't know if this is true of all species, but at least some of them have this extreme sexual dimorphism. What you're looking at here is, I would assume, a female. The males are really, really, really tiny. And their mating process is they find a female and they burrow into her skin and live there permanently. And she provides the nutrients and he provides the sperm, and that's their
It's
relationship.
amazing. The males
Yeah.
are parasitic.
Yeah.
Yes. Wait, so Jolene, the size.
I thought a lot of you might appreciate
I'm
that.
just saying, in
We
this
already knew
case,
that, no?
in this case, I'm not
Yes.
saying that. In the case of the anglerfish, so yeah, these Jolene, they're about 8 inches, so like 20 centimeters. The females, and as Alan said, the males are like 1 to 3 centimeters, so like an inch. They're tiny, so it's probably like
Still,
this
I think
big.
it could fit my arm in its sharp-toothed mouth.
Probably. It's mostly mouth. Yeah.
But if you're ever scuba diving that deep, you're dead anyway.
True.
Brianne, what do you have for us?
So I have a book that I read a while ago called Moonwalking with Einstein, The Art and Science of Remembering Everything. And this book was written by a journalist who learned about something called the United States Memory Championship, and basically in the United States Memory Championship, the competitors have to memorize the order of cards in a deck, and see who can remember the most cards in the shortest amount of time. And the author then starts training with these competitors, and in fact wins the memory championship and learns sort of all about techniques for memory, and just generally talks about what we know about memory. Also talks about some individuals who are unable to remember and sort of what was going on there. And I remember reading it and finding it just really fascinating, and trying to use some of the techniques for, 5 minutes or so. And it felt like they didn't seem to link in with my brain, but I just thought the whole thing was really fascinating. And the reason why I was thinking of this is that this week, there was a article in the Times about a World Memory Championship winner who had memorized 80 numbers in 13.5 seconds. And so I read about that. It was a 20-year-old who had to memorize these numbers. And he was talking about his process and how he learned how to do this. And everything that was discussed when I read that article reminded me exactly of what I read in Moonwalking with Einstein. It's a lot of the same techniques that I'd heard much more about, explained in a lot more detail. So I thought that was really interesting. Basically, the idea is that each number or each card or whatever you're remembering, you assign a something else to. You assign like a person, or you assign an action, and you imagine them in a room that you're walking through. And so then you just picture it and think about what you would see in order. And so that's where the idea of Moonwalking with Einstein comes from, is that the author was trying to remember some cards, and one of them was somehow related to moonwalking, and one of them was somehow related to Einstein. And so by remembering the idea of Moonwalking with Einstein, he could remember those cards going together.
That's so cool. I actually just bought it on Amazon as we were speaking.
I just added it to my library list.
You've convinced me.
I found it really, just an interesting out of the blue thing to read.
Yeah.
Cool. Kathy, what do you have for us?
I picked Witness History, which is a BBC show. And for us here, it comes on our radio station at the end of the BBC feed at 4:50 a.m. And sometimes I wake up in time to hear it. The other day, I woke up in time to hear just part of it, so I had to go back and find the rest of it. But you can use this link to get to these 9-minute short podcasts about all different things in history. Sometimes they're an interview with the person who was there, sometimes not. So recently, Paul Keating's Redfern speech caught my eye. It's one of the top 3 speeches ever, according to someone's ranking. You can see what the other 2 are. So they're just really fun, all different kinds of things in history. And I really like listening to them. So The World's Longest Kiss is the one I woke up in the middle of. And so it's like, what?
And Eisenhower's farewell address is on this list. That's the Cross of Iron speech, right?
I don't know.
I was wondering if it has the military-industrial complex.
Yes,
that's
it does.
The Cross
it
of
does.
Iron speech.
I did hear the military-industrial complex, and that was it.
That
You're
is
right.
one of the great speeches.
Yeah.
Cool.
Yeah, check it out. And you can also download the whole BBC Sounds thing and find lots of other cool things. Another one of my favorites is the Cultural Hour with Nikki Beatty. She's amazing. So that would be fun. She does movies, TV shows, all kinds of interviews with great people.
Cool. Rich, what do you have for us?
I have another pick from the astronomy picture of the day, which I have to give a nod to Kathy because she's the one who turned me on to APOD, which is my homepage on my browser now. So
Me too.
I see
Me
them all
too.
the time.
Thanks, Kathy.
It's in my
Thank
feed.
you.
Yep.
I saw this and did what I do. I bookmarked it as a TWiV pick without really much other thought about when I would use it. And then I looked at it and I thought Dickson, okay, because Dickson would love this. It's really a mashup of Dickson stuff. This is a video that is a flyby of Jupiter by the Juno spacecraft, which the Juno spacecraft was launched specifically to study Jupiter and makes orbits that are a highly elongated orbit. I don't know why. That has a 53-day period, but on one end of that, it flies very close to the surface. And this is a video of one of those flybys. This it arrived in 2016. I don't know if this is still going on, and I don't know exactly when this video was taken. I'd have to dig a little deeper, but this is a video of a particular flyby. That's one of the highest resolution images or series of images of the surface of Jupiter. And it's just absolutely stunning. And they have set it, so it's I'm channeling Dickson here. It's scientifically amazing. The images are artistically amazing. And in fact, to me, they're very Van Gogh-ish. All right. I see Van Gogh throughout this. And they've set it to Beethoven's Moonlight Sonata, which really
Yes.
tops the whole thing off. So I offer this channeling Dickson for everybody to just enjoy. It's amazing.
And Juno had a planned mission of 7 years. It is now still running after 13 and a half years.
Okay, thank you.
Typical NASA
Cool.
type of thing.
Typical NASA stuff, yeah.
Nice. Alan, what do you have for us?
Yeah, so I also picked a pick that I think I've had this sitting there for a little while, and I thought, yeah, I've got it. I had actually kept it in the queue because I was hoping to pick it when Dickson was on.
Mm.
So here we go. This is an article. It's a long article in Science. A reporter, Dan Charles, went to the field to this site in Bulgaria where a researcher is studying what happens to farmland and farming towns after the land is abandoned by the farmers.
Hmm.
And it's a really interesting problem because, there's long been this assumption that, well, farming is an artificial use of the land. If we abandon the farmland, it'll go back to whatever it was before. And a lot of that has been based on research that was done in the northeastern U.S., especially in New England, where we had my part of the state, western Massachusetts, used to be all farms, and now it's hardwood forest for the most part. And so that happens in some ecosystems, but it turns out around the world, as you abandon farmland, all kinds of different things can happen. It depends on what invasive species are available to populate it, and how it's abandoned and what it was used for, and what the terrain is like. And of course, this is all changing with climate change, but it's an interesting dive into a complex, nuanced problem that I think most people haven't given a lot of thought to.
Yeah, Dickson always used to talk about moving the farms into the buildings, and then the farms let them grow back to forests, right?
Which can work, but it turns out it doesn't automatically work. It may or it may not, and this researcher is working on figuring out what the factors are.
It's cool. Jolene, what do you have for us?
Yeah, so I decided that I wanted to pick the the lectures that Dickson has on parasites. So when I was finishing up in graduate school, I had the opportunity to teach medical microbiology, which had traditionally been taught as basically bacterial pathogenesis at the molecular level. But I had been listening to TWiV and TWiP and so forth for a while. And so I watched all of Dickson's lectures that were available at the time. And in my course, we talked about viruses and parasites and bacterial pathogens and even archaea. And so I learned all the parasitology, pretty much most of it, from Dickson that I know. And his lectures were fascinating. I also recommend the first 27 episodes of This Week in Parasitism, the podcast, because Dickson tells great stories and they're very memorable. I still think about his stories, which are more like kind of examples of ways that toxoplasma could be spread between cats and humans, and how, your cat, like Alan's cat right now, they leap up where you are. And so if they come when you're in the kitchen and you feed them a little bit of steak, then you can be spreading toxo and around. And then he would say, but if you freeze it, then this won't happen. And so yeah,
Freeze the steak, not the cat.
Exactly. That's right. Freeze the steak. So I really enjoyed all of those lectures. I'm not sure that the versions that I watched are the ones that I've linked to here, but this is, I think, a redone version with Dickson and Daniel for a whole long series of lectures. And yeah, for your enjoyment.
So I originally recorded those videos in my office at Columbia, and I sat Dickson at that table and put a green screen behind him, with the idea being, and then I used it actually to put images behind it. But I remember on multiple occasions falling asleep, and I said, gee, this is how Dickson feels when he's falling asleep on a podcast. It was so funny. And he would stop and say, Vincent. I think the video should be the same, Jolene.
Okay.
Because I didn't do any editing.
I see.
As far as I know. So
awesome. Well, they're great.
That's cool. My pick is an interview I did some time ago when Dickson's book was published, The New City. So I brought him here to the incubator and sat down with him. And there it is. I think it's a nice interview. And that's the last interview I did. Did I do any others? I think I didn't. I think that's the only one. So you could see him talking about a little bit about himself, but mostly about the book. I went through the different chapters in the book and had him explain it. It was cool.
Cool.
Which, once again, if you read one Dickson Despommier book, then I would pick that one just to
New
get
City?
a sense of him. Yeah, The New City, I think it really crystallizes a lot and kind of gives you an impression of how he thinks.
He has one other book that he published, People, Parasites, and Plowshares.
Yes.
Mm-hmm.
It's about parasites,
Right.
In a folksy way, not a technical way. So that's also very
That's
interesting.
another good one.
All right, that's TWiV 1195. You can find the show notes at microbe.tv/TWiV. Questions, comments, picks, send them to twiv@microbe.tv. And we would like to have your support to continue these programs. Microbe TV is a 501c3 nonprofit, which means in the U.S., your donations would be federal tax deductible. And you can go to microbe.tv/contribute for the various ways that you can help us out. My first entry here for years, as long as we have been doing Dickson Despommier, is at trichinella.org and thelivingliver.org. I'm going to just leave that there. Graham Barker's at Drew University, BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot, and also I miss Dickson.
Yeah, it's been a long time already since he's been on a podcast. Kathy Spindler is Professor Emerita, University of Michigan in Ann Arbor. Thank you, Kathy.
Thank you. I think it was really good that we got to all talk about different things about Dickson, but even this doesn't capture everything. So, thanks.
Oh, there are lots of videos you can go to and see more. Yeah.
Right.
I'd love to go through all of them, pick out the most interesting moments, right?
The most Dixoniest moments.
Yeah. Well, I have so after we did a podcast in Hamilton, Montana, and after that, he insisted on taking me on a fishing trip, a float, he called it, on the Bitterroot River. So we did, and I watched him fish for 6 hours, and I have most of it on video. So one day I have to edit it and release it because he's a just fabulous fisherman. He caught trout after trout after trout, and each one he would put in the net, take it out, he would kiss it, take the hook out, and release it. He kissed every fish that he caught. It's really interesting. One day I have to put that up. It's a lot of fun. Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Ciao, Dickson. See you when the sun shines.
Angela Mingarelli is at McGill University Immune Vet on Blue Sky. Thanks, Angela.
Thank you so much. Yes, Dickson, I feel like his essence will always be in TWiV. He'll end also in our hearts. But yes, we definitely miss Dickson.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure. Farewell, Dickson.
Jolene Ramsey is at Texas A&M University and ramseylab.vercel.app. Thank you, Jolene.
Yeah, I just want to say thank you to everyone here. I never took the opportunity to say thank you directly to Dickson, but I'm really grateful for being part of this all-star team of science educators and communicators. So it's been a blast, and I'm glad that we continue to carry on the legacy that you started with Dickson a long time ago.
I'm Vincent Racaniello. You can find me at microbe.tv. We're also on Blue Sky, Microbe TV, no dot. And Dickson, I'll miss you. It was a lot of fun doing this with you. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.